All right, it is 12 o'clock, so I'm going to go ahead and get us started right on time. Welcome to Medical Grand Rounds. Appreciate everyone being here. We are filling this auditorium up more by the week, and I like that. A little bit of a teaser for folks who are thinking about coming in the next several weeks. On November 15th, we're going to welcome our own Dr. Joyce Lee. She's going to talk about autoimmune interstitial lung disease and particularly advances in RA-associated ILD. And then just as a reminder, on November 22nd, we are off for the Thanksgiving holiday, so do not, this is the opposite of what I say every other week, do not come here on November 22nd. But we will kick things back off on the 29th with Professor Mei-Lan Han from the University of Michigan, who's going to talk about the Achilles heel of lung health and her work in COPD. I'm going to turn all of our introductions today for our special Gill Lectureship over to Dr. Eric Pershla, who's the Division Director for Infectious Diseases. Dr. Pershla.
So thank you very much, Jeff. Welcome everyone to Grand Rounds. It's a great pleasure for us to welcome Dr. Dan Baruch, who is our speaker today, and also the 2023 Tim Gill speaker. Before I introduce him, I want to emphasize that we're deeply grateful to Tim Gill, who endowed this annual lecture series 30 years ago. And it was intended to bring distinguished leaders in HIV research to come here and tell us about their work. And Tim is the founder, that's Tim up there at the top. He is the founder and co-chair of the Gill Foundation, which is a Denver-based philanthropic organization supporting efforts to secure nationwide civil rights for LGBT Americans. Tim is actually the single largest individual donor to the LGBT rights movement in US history. And they presently concentrate on countering discrimination based on sexual orientation and gender expression. They're very active and effectively so in electoral politics in the United States and multiple state elections and federal elections. And they've been a major factor in advancing acceptance and human rights for the LGBTQ community across the nation. So Dan is our distinguished 2023 speaker, and I want to say it's both timely and right, just right, that someone with his research interests should give this lecture. A bit of perspective for this audience: Tim endowed this series 30 years ago during the most really desperate times of the HIV pandemic, before we had even protease inhibitors, before we had triple combination therapy that's now become so highly effective and has been condensed into extremely convenient single pill regimens that really work. But at the same time, we know that there are a lot of impasses and complications, and we know that we can't treat our way out of the pandemic. And so the field is focusing a lot on two things: definitive cure and an effective vaccine, both of which are very, very profoundly difficult goals. Dan has been active in both of those areas, and he'll tell you about that today. So the thing switched, and I don't even know how to. Oh, there we go. Okay. Dan graduated from Harvard College, received his PhD in immunology from Oxford University, where he was also a Marshall Scholar, his MD from Harvard Medical School in 1999. He was a medical resident and then an ID fellow at MGH and the Brigham, respectively, and then came on faculty there in 2004. He is currently the William Bosworth Castle Professor of Medicine and Professor of Immunology at Harvard Medical School. He's also a member of the Ragon Institute, and he's director and founder of the Center for Virology and Vaccine Research at Beth Israel Deaconess Medical Center. He's been the recipient of an incredible number of prizes and awards. I'll just mention a few: the Maxwell Finland Young Investigator Award, the IDSA Oswald Avery Award, the King Faisal Prize in Medicine. He's been elected to the National Academy of Medicine, and the World Vaccine Congress has given his team the Best Academic Research Team Award. But most interestingly, in addition to being the Boston Globe's Bostonian of the Year in 2016, he received the 2021 Boston Celtics Heroes Among Us Award. He's here receiving that in front of 20,000 screaming Celtics fans on opening night in the Boston Garden. And he's also received the Medical All-Star Award from the Boston Red Sox. So he's led the development of vaccine candidates for multiple pathogens, HIV, Zika, SARS-CoV-2, which he spoke about this morning. He's pioneered adenovirus, Ad26 vectors in particular, novel immunogens. They developed a SARS-CoV-2 vaccine using the Ad26 platform that eventually became the J&J vaccine, which has been given to millions and prevented many, many deaths worldwide. A lot of work in non-human primates on HIV, on cure strategies, a lot of work on eradication strategies. I could go on and on. He has 8 NIH and R and U series grants, and he's published 428 peer-reviewed publications. So his talk today will be novel HIV vaccine and cure strategies. Dan Baruch.
I'm going to see if I can. I don't know if I can. I think someone can. You have a career in IT. That's right. Thank you.
Well, thank you, Eric, for that very kind introduction. And it's my pleasure, privilege being this year's Tim Gill visiting professor. I was told to show this slide first. So over the next 45 minutes or so, I'd like to give you an overview of some of our work that's really spanned quite a number of years now on HIV vaccine and cure strategies. And I'll say that unlike SARS-CoV-2, where we and others were able to develop vaccines within 1 year, then the world has not been able to develop an HIV vaccine in 40 years. And I'll try to give some explanations as to why this is the case and perspectives. So here are my disclosures. So the history of humankind is intimately intertwined with the history of pandemics. Starting from early recorded history, from ancient Greek times, the plague of Athens, to the Roman Empire, the plague of Antonine and plague of Justinian, to the Middle Ages and the Black Death. In the 1500s, the plague of the Incas. In the 1600s, the Great Plague of London. In the 1700s, the yellow fever pandemic. In the 1800s, the cholera pandemic. In the early 1900s, the Spanish flu pandemic. And over the last 100 years, sequential pandemics of Ebola, HIV, avian flu, SARS, chikungunya, Zika, COVID, monkeypox, and others. I can't tell you when the next pandemic will strike or where it will happen or what it will be, but I can tell you there will be more pandemics. And the voice of Stanley Plotkin, who's one of the foremost vaccinologists of our time, in the first edition of his textbook, he writes, "The impact of vaccination on the health of the world's peoples is hard to exaggerate. With the exception of safe water, no other modality has had such a major effect on mortality reduction and population growth." It's really a deep and astounding statement, which I agree with wholeheartedly, that in all of clinical medicine and public health, everything of what we do, the biggest impact we've ever been able to have is safe water, and secondly, vaccination. So classical vaccine strategies involve the use of live attenuated organisms, whole killed organisms, and protein vaccines, including toxoids, protein conjugates, and protein subunits. Over 20 years, but really solidified during the 3 years of the pandemic, have been the evolution of a new series of vaccines called gene-based vaccines. Essentially, these are vaccines that don't require the actual pathogen, but just require the target antigen. such as the COVID spike, maybe an HIV envelope. These are nucleic acid vaccines, such as DNA and mRNA vaccines, and viral vector vaccines, such as adenoviruses and poxviruses. This is from a review article that I wrote in 2021 with colleagues from Moderna, in which we really tried to draw conceptual parallelisms between nucleic acid and viral vector vaccines. They're essentially conceptually very similar. You just give a target antigen's nucleic acid, DNA or RNA, with a delivery vehicle. For mRNA vaccines, it would be the lipid nanoparticle. For vector vaccines, it would be a viral capsid. The goal of the delivery vehicle is to get the nucleic acid into cells. Once it's in cells, then it generates the protein and delivers an immune response. So HIV and SARS-CoV-2 are two contrasting global pandemics of our time. HIV has been with us for over 40 years. It's a retrovirus, causes chronic persistent infection, immunodeficiency in AIDS and untreated individuals, over 36 million deaths, enormous viral diversity. The envelope protein is notoriously difficult to neutralize, and despite 40 years of concerted global research, no vaccine is available. SARS-CoV-2 has been with us for about 3 years. It's a coronavirus that causes an acute infection, respiratory illness, COVID-19. It's believed to have caused about 15 million deaths. There is limited viral diversity, although with variants that's changing. The spike is relatively easy to neutralize, although it's changing with variants, and multiple vaccines are available. And in fact, multiple of the technologies used in the HIV and developed in the HIV research field were employed for SARS-CoV-2 and really facilitated the development of vaccines multiple vaccines in just about a year. If it were not for HIV research, and the HIV researchers, basic and clinical, who actually were many of the SARS-CoV-2 researchers, if it weren't for the investment in HIV research, we would not have had the COVID vaccines, certainly not in the time frame we did. So in 2011, there was widespread prediction for the end of AIDS. It was predicted that by the year 2020, new infections would go down to about 500,000 people and would go down to zero shortly after that. So what happened? Clearly this did not occur. The reason is that despite tremendous advances in HIV therapeutics, where we can now control virus in the vast majority of people for seemingly an indefinite period of time, oftentimes with a regimen as easy as one pill once a day, there's been much more limited progress in prevention. And while treatment is life-saving for those who are infected, then we likely can't treat ourselves out of the pandemic. And we have to improve our ability to prevent infection. So this is, according to UNAIDS, adults and children newly infected with HIV from 1990 to 2018. And you can see there is progress. There is a decline in HIV prevention, but it's slow and painful. And according to the most recent numbers, from UNAIDS in 2022, there were still 1.3 million new infections and over 600,000 HIV deaths. A truly astounding number, particularly since HIV has largely fallen out of the mainstream media. But it is widely believed by a number of influential authors, including this one, that an HIV vaccine is essential for ending the HIV/AIDS pandemic. So writing this article in 2017, and really similarly as of today, Tony Fauci and many other people feel that despite the remarkable gains in treatment and prevention of HIV infection, the development of an effective vaccine will likely be necessary to achieve a durable end to the HIV pandemic. It's really only vaccines that have ended pandemics. Treatment is fantastic and life-saving for those who are fortunate enough to receive it, but treatment has never been able to eradicate viral pathogen. However, the scientific challenges in the development of a prophylactic HIV vaccine are unprecedented in the history of vaccinology. This is a list that I put together as a table in a review article I wrote 15 years ago, and I will say that there have been major scientific advances in each one of these points. However, these still remain the main scientific challenges 15 years later. Unlike pathogens for which we have made vaccines for, such as smallpox and to some extent SARS-CoV-2, for HIV, there is extensive viral clade and sequence diversity. It's been said that the sequence diversity in a single HIV patient is greater than the sequence diversity of global influenza, just for perspective. Also, there is very early establishment of latent viral reservoirs. So a vaccine will have to act really quickly to prevent permanent infection. The immune correlates of protection are unclear. And this morning I gave a talk on SARS-CoV-2 vaccines and how the early identification of immune correlates really facilitated vaccine development for SARS-CoV-2. For HIV, the virus can mutate rapidly to evade both humoral and cellular immune responses. And unlike SARS-CoV-2, for HIV, there's no method that exists today that can elicit broadly reactive neutralizing antibodies, which is probably one of the key hurdles for making an HIV vaccine. Attenuated viruses are simply too unsafe for human use, and there is very little work in this direction for HIV. So over 40 years, there have really only been 5 HIV concepts that have been tested in humans for efficacy. So just think about that. For a problem over 4 decades, research ongoing in the entire world, only 5 concepts have been tested for whether they work in humans, which is, in my view, not nearly sufficient for the magnitude of this problem. Starting from the early Vaxgen GP120 vaccines that aim to induce humoral immunity, these simple vaccines were able to raise immune responses, but in phase 3 studies in US and Thailand in 2003 showed no efficacy. The Merck Ad5 Gag-Pal-Nef program, the goal was to raise cellular immunity, and phase 2B studies in the Americas and Africa also showed no efficacy. Then a glimmer of hope in the field was a prime boost strategy that involved a Sanofi canarypox vector called Alvac, followed by the same protein boost made either by AIDS, Vax, or Novartis, GP120, that essentially was used in the Vaxgen studies. The goal was to generate humoral and cellular immunity. And while the RV144 trial in Thailand did show a significant 31% protection in 2009, a follow-up study that basically used a similar concept did not show efficacy in Africa. And the NIH Vaccine Research Center program that also had a prime boost strategy of a DNA prime Ad5 boost in a phase 2B study showed no efficacy in the United States in 2013. And then a study, a program that we were very involved in was the Janssen Ad26 Mosaic Prime GP140 boost, also aimed to do a little bit better in generating humoral and cellular immunity. And unfortunately, this vaccine also did not show any substantial levels of efficacy in a phase 2B study in Africa called Imbokodo and a phase 3 study in the Americas and Europe called Mosaic. But I would say that despite none of these truly being a home run hit that would lead to marketing of an HIV vaccine, nevertheless, I think the field has learned a lot through all of these programs. So this is the canarypox GP120 vaccines that were initially showed some promise in a Thailand study with a 31% efficacy shown on the left. But a subsequent follow-up study in South Africa really showed no efficacy at all. When I started my lab in 2002, then we reasoned that we needed more vaccine technologies. And so we developed a series of adenovirus vaccine vectors. At that time, Merck and the NIH were developing Ad5 vectors for HIV vaccines and other things. And we reasoned that Ad5 would not be an optimal vaccine vector because the vast majority of people, particularly in Africa, not only had baseline immunity to Ad5, but had sky-high neutralizing antibody titers that would be suppressive. So we developed a series of adenovirus vectors from other serotypes, and this is our first paper in 2007, in which we developed 6 new adenovirus vectors, including one that was called Ad26, serotype 26. And so we developed Ad26 as a vaccine vector platform for a variety of diseases and partnered with industry for that. The features of this platform is that it induces robust antibody and particularly good CD8 T cell responses. Another feature of this platform is, unlike mRNA vaccines, it gives durable responses for more than a year. And also, it is very good for developing world applications because there's no freezing requirement and it's stable in liquid form. So it's much more deployable as a global vaccine. In the SARS-CoV-2 pandemic, there was a very extremely rare 3 in a million thrombosis complication, which might limit the use of this vaccine in the Western world. It seems like it's really not a complication in the developing world or with re-administration. So we actually think that using a lower dose of this vaccine would likely overcome this problem, or a different serotype that would not have the platelet factor 4 binding issues. But the Ad26 Ebola vaccine has been licensed and is one of the 2 Ebola vaccines currently being deployed in Central Africa. The Ad26 COVID-19 vaccine eventually became the J&J vaccine that's been licensed for emergency use and it's been given to somewhere between 200 and 500 million people throughout the world. And there have been phase 1, 2, and 3 studies for RSV, HIV, Zika, and other pathogens. So the HIV vaccine strategy that we pursued for a number of years really involved the premise that we need vectors to elicit optimal immune responses. We needed a strategy of antigens to achieve global coverage that we called mosaic antigens. And then we used purified proteins to try to augment humoral immunity. So this was based in part on a series of non-human primate studies in which we showed that priming macaques with an Ad26 vaccine expressing SIV-ONV GAG-PAL and then boosting with a purified SIV-ONV protein gave better protection than vectors alone. So you can see the ad protein approach is shown in blue, the vectors alone is shown in red, and the sham controls are shown in black. And here, the way the studies are done is macaques are vaccinated and then are given a series of 6 intrarectal heterologous SIV MAC251 challenges, and the percent uninfected animals is shown on the y-axis, and the number of challenges is shown in the x-axis. So you can see all the sham controls get infected, the vectors alone displace this curve slightly to the right, and we get better protection with the blue curve, the Ad26 prime with a protein boost. And this in this study was about 50%. But I should say that there's clear degradation of this with further challenges. So if we were to challenge these animals 10 or 20 times, we would likely degrade this protection. So this was coupled with some work that we did with bioinformatics with Betty Korber from Los Alamos, in which we developed optimized immunogens to try to cover global diversity. It's an interesting, essentially, math problem. How are you going to develop a discrete set of vaccine immunogens that would best cover a variable pathogen? Now, if you deal with smallpox, that's a single sequence, pretty easy which sequence to use. You just identify what the target antigens is and use that sequence. For HIV, then, and maybe to some extent, the same concepts are being applied now to a pan-coronavirus vaccine. But for HIV, the diversity is so great. How are you going to even approach developing sequences? So we worked together with the bioinformaticians at Los Alamos and developed a computational strategy to develop so-called mosaic antigens that at the very least would do better than any single sequence antigens. And in macaques, we showed a proof of concept in 2010 that the mosaic antigens shown in blue did raise increased breadth of CD8 and CD4 T cell responses. And in humans, although this was not a head-to-head study, then the studies with the mosaic antigens in blue showed about a 10-fold higher epitope coverage than the prior Merck vaccine, which was a single antigen vaccine. So the vaccine that went forward into clinical testing involved priming at week 0 and 3 with an Ad26 vector expressing GAG-PAL and ONV inserts, and then boosting at month 6 and 12 with the vectors together with proteins. So 2 efficacy trials were done, one called Imbokodo, which was a phase 2b study in 2,600 young women in sub-Saharan Africa, and a phase 3 study called Mosaico, which was in MSM and transgender individuals in the US and North America and Europe. These differed in terms of the one being clade B, one being clade C, the genders of the participants, the route of exposure, and the nature of the protein boost. So unfortunately, in the Imbokodo study, as well as the Mosaico study, the primary analysis showed vaccine efficacy was not significantly different from 0. There was a hint of a trend towards a low level of protection, but certainly not something that was significant or something that could be used for marketing moving forward. We have learned from this study, and it appears in correlates analysis that there is a trend towards protection in individuals that had higher levels of IgG3, V1, V2 responses, which was actually the correlate of protection seen in the original RV144 study from Thailand. However, this is, of course, a post hoc analysis, and so this needs to be viewed as a hypothesis-generating result rather than a definitive result. But as I mentioned in the beginning, the HIV vaccine research as a field, as well as our program specifically, had a very important effect on the pandemic that started 3 years ago, which was COVID-19. And so I'll just give an example of our own research and how it impacted COVID, which is that the generation of this vaccine platform, the ad-based vaccine platform for HIV, directly led to a series of ad-based vaccines that were used for COVID-19, including the Ad26 specifically, which became the J&J vaccine, as well as the ad vectors, the chimp ad vectors that became the AstraZeneca vaccine. And for the COVID-19 vaccines, then these were highly successful, were developed very rapidly and deployed throughout the world. And as a whole, the COVID-19 vaccines are estimated to have saved at least 20 million lives. And I'll mention that the ad-based vaccines from AstraZeneca, J&J, CanSino, and Sputnik actually saved more lives in 2021 than the mRNA vaccines from Pfizer and Moderna, although all in totality is what really led the impact on the world as a whole. But with the COVID-19 vaccines, I think we have to also acknowledge the global health disparities that emerged in the rollout. That in 2022, when over 70% of people in the United States were vaccinated, and we actually had sufficient vaccine to vaccinate 100% of the population with multiple boosts thereafter, even boosts that people didn't want to get, then still less than 15% of people in Africa were even offered a single shot of any COVID vaccine. And this global health disparity is something that is curious. I find that most doctors and scientists in the United States really don't appreciate just the extent of this global health disparity that was driven by the inequitable distribution of the vaccines. And it's been estimated that if there was a equitable distribution of these vaccines globally, then mortality rates in the Western world would not have been worsened, and we would have saved over 600,000 lives in Africa during the year 2021. And the global health disparity is really driven by the fact that the mRNA vaccines, as outstanding and successful as they were very difficult to transport, largely because of their cold chain freezing requirement. It's really hard to get a vaccine that needs to be at minus 80 degrees in a town in the middle of Africa that has no freezers at all. So there was very limited use of mRNA vaccines in Africa in 2021. And the workhorse vaccines in Africa in 2021 were the ad-based vaccines from AstraZeneca primarily, and J&J to some extent. And because we had a role in the development of this vaccine, one of the most humbling moments we had in the lab was the first COVID-19 vaccine rollout in South Africa, which was the J&J vaccine on February 17th of 2021. This is at a time in South Africa when the COVID-19 pandemic was raging, and there was essentially nothing to offer. And the mortality rate amongst healthcare workers in South Africa was astronomically high, with very high mortality rates of doctors and nurses and other healthcare professionals, which was really threatening the healthcare infrastructure of South Africa. And so this was the first vaccine rollout in South Africa. I believe it was the first vaccine rollout anywhere on the continent of Africa, and it was just 3 weeks after the phase 3 global efficacy results were announced. So just imagine the pace of it, that 3 weeks after the first announcement of interim efficacy in a phase 3 study, it's already being rolled out in a population on the continent of Africa. And here, our colleagues, Slim Abdul Karim and Qureisha Karim and Nigel Garrett, who actually were our collaborators for our clinical HIV vaccine research in South Africa, became our collaborators for COVID-19 vaccine research in South Africa. And here they're personally overseeing the first shipment of COVID-19 vaccines to South Africa, and announced on Twitter, like most things during the pandemic. Nigel writes, immensely proud to deliver the first COVID-19 vaccines to healthcare workers in KwaZulu-Natal, Prince Mashani Memorial Hospital. Let's accelerate this effort and save lives. On the right, you see a picture of a healthcare worker getting vaccinated and the president of South Africa getting vaccinated on February 17th. And the result of this early rollout at a critical moment in time, the demonstration project that was followed as a phase 3B demonstration project was called Sasanki. And a single shot of this vaccine resulted in an 86% protection against death in South Africa healthcare workers. And it's really rare in a clinical research article to use death as an endpoint of large numbers of healthcare workers. It's really astounding and really, almost it's almost difficult to even imagine, because at that point in time, there were no other options. And so this was the only vaccine option and really only prevention option that was available, but had a massive impact on saving the lives of healthcare workers and subsequently the general population for the rollout. So back to HIV. So what do we learn from the Ad26 envelope HIV vaccine? So I think this was an example of a successful academic industry government partnership that brought a vaccine from concept and design up through phase 3 efficacy trials. Unfortunately, Although it was safe and well tolerated, it did not show any substantial levels of efficacy. We were able to expand cellular immune breadth with the mosaic immunogens, but this was likely insufficient for global HIV diversity, even with the mosaic antigens, and we also were not able to induce broadly neutralizing antibodies. No other HIV vaccine is sort of in the pole position, and there's no other HIV vaccine that I'm aware of that'll be ready for clinical efficacy trials at any point in the next few years. But the vector platform was able to be used to develop successful Ebola and COVID-19 vaccines that were marketed and have been estimated to save at least 1 million lives globally. So HIV vaccines looking forward, where do we go from here? So I would argue that testing 5 concepts in 40 years as a global effort is simply not enough. I mean, for a problem of this magnitude and scope and duration, then we need to test more than 5 things. We need to test more than 1 a decade. So there is really continued basic HIV research, as well as, until we have a vaccine, non-vaccine strategies for HIV prevention. So I'll say a few words about each one of these. So for continued basic HIV vaccine research, we and others are trying to develop envelope immunogens to induce broad neutralizing antibodies and strategies to induce better effector T-cell responses. So the current lead concept in the field is so-called germline or lineage immunogens. How can we design an immunogen that induces broadly neutralizing antibodies given the vast degree of global virus diversity? This is currently an unsolved problem, and no strategy as of today has been successful in inducing broadly neutralizing antibodies for HIV. The lead concept in the field now is so-called germline priming, which is to trigger the germlines of known monoclonal antibodies with sequential boosting of immunogens that essentially help you draw a B-cell ontogeny according to a known pathway that leads to broadly neutralizing monoclonal antibodies. This is a tall scientific order. Nothing like this has ever been attempted before in any field. Germline priming has been able to be reported in humans, but so far there's no proof of concept that shows that this strategy will lead to induction of broadly neutralizing antibodies in either macaques or humans with the breadth, magnitude, and durability to be clinically relevant. And even if such a strategy becomes scientifically feasible, then it may require a complex boosting regimen that would be difficult to implement. If we've learned one thing from COVID, is we need to have a regimen that is simple and deployable. So an alternate HIV vaccine concept is using epitope-focused immunogens. Epitope-focused HIV envelope vaccines have received less attention, but in my opinion, may offer an alternative strategy for the induction of broadly neutralizing antibodies. Epitope-focused envelope immunogens involve protein engineering to increase exposure of broadly neutralizing antibody responses to certain epitopes. And the key epitopes are either the fusion peptide, the V2 epitope, the V3 epitope, or the CD4 binding site. Those are really the 4 known broadly neutralizing antibody epitopes on an HIV envelope. The concept that's probably furthest along is a fusion peptide immunogen from Peter Kwong at the NIH Vaccine Center that has been able to induce low magnitude but clear tier 2 neutralizing antibodies in non-human primates and is currently in phase 1 clinical trials. These epitope-focused immunogens can be developed as standalone vaccines or as boosters after germline immunogen priming. And also several of these epitope-focused immunogens can theoretically be combined to induce neutralizing antibody responses against multiple epitopes. So an early phase program that we've been involved in involves a vaccine strategy that we've called signature-based epitope-targeted or SET vaccines. This is a collaboration with Betty Korber, in which we have used bioinformatics to analyze V2, V3, CD4 binding site, and membrane proximal external region sequences in HIV viruses worldwide to develop HIV immunogens with improved exposure and diversity coverage of key BNAB epitopes. So we initially developed V2 optimized set-on vaccines, given this clear signatures of V2 specific antibodies in terms of sensitivity and resistance that I'll show you in the next slide. We built immunogens on a clade C primary isolate 459C envelope backbone that had some intrinsic stability and was able to elicit modest levels of antibodies in guinea pigs. And then we developed a V2 set immunogen cocktail, which was a trivalent cocktail of wild type immunogens, as well as modified immunogens that either enhance epitope exposure or incorporate relative epitope diversity. So the global sequence analysis basically allowed an analysis of these 4 key antibody targeting regions: the V3 glycan region, the V2 glycan region, the CD4 binding site region, and the membrane proximal external region. We developed these trimers and show that they are high quality trimers by electron microscopy. And by antibody binding, we show that they exhibit this, the expected patterns of binding to the relevant monoclonal antibodies. In preliminary studies in guinea pigs, we showed that the V2 set immunogens, shown in the bottom, gave better neutralizing antibody responses that could be achieved with either the parental version or other leading single immunogens of the time. And the diagrams, let's see if I have a pointer. The diagrams here show you the positions of the mutations that we engineered. So we then put these forward into a macaque study, and naturally occurring enveloped GP140 immunogens typically do not raise heterologous tier 2 neutralizing antibodies in either macaques or humans. Certain strains, for the aficionados in the audience, BG505 and other SOCIP stabilized GP140 immunogens can induce autologous antibodies against the exact same strain, but these responses are often directed against glycan holes and the exposed basal surface. In other words, artifacts that really don't show any relevant breadth. So our goal here was to assess if this new vaccine strategy could induce heterologous tier 2 neutralizing antibodies, and if so, whether the antibodies would be directed against V2, which is the target epitope. So this is a collaboration with Peter Kwong, and we also looked at whether the combination of our V2 directed immunogen with his fusion peptide immunogen could induce antibodies against both targets. So this is a macaque study. It's not yet published, but we're writing it up soon. That really had 3 groups. The first group just received the wild type 459C envelope immunogen. The second group received the V2 set modified immunogen with the fusion peptide. And the third group just received the V2 set modified immunogen. So here are binding antibody titers showing the induction of both fusion peptide antibodies on the left and envelope antibodies on the right. And they give you the expected patterns that the groups that receive the fusion peptide in groups 1 and 2 had the highest fusion peptide responses. And all of them, all 3 groups generated envelope titers, with the ones that received the optimized trimers, the green and the blue, being slightly higher. If we look at neutralizing antibodies, then all 3 groups raised what we call tier 1 neutralizing antibodies, which are easy to neutralize viruses and probably not very clinically relevant. And the most clinically relevant assays are these, which are the heterologous tier 2 antibodies against essentially patient isolates. And here I'm showing 3 hits that we had. Unfortunately, it's 3 out of 20, but I'm showing you the 3 that actually worked. And we were able to show that the modified immunogen, the V2 set immunogen, but not the original immunogen, was able to generate neutralizing antibody titers at least to a subset of tier 2 viruses. And in studies that Peter Kwong did, the VRC, he showed that if we mutate the V2 epitope to create this K169E mutation, then we're able to show that the neutralization goes away, thus demonstrating that the majority of the reactivity actually is against the V2 epitope, which was the target of this antigen design strategy. So this is still at a very early stage and very basic, but the set-on vaccine strategy aims to increase epitope exposure and epitope coverage to improve heterologous tier 2 neutralizing antibody responses. Our data show that the V2 set on vaccine increased heterologous tier 2 neutralizing antibody responses in NHPs, and that these antibodies were primarily directed against the V2 epitope, consistent with the design concept. Additional boosting is in progress to determine whether the heterologous tier 2 antibodies will further increase in scope, and expanded neutralization and structural studies are currently in progress as well. So this is a very early concept that we're pursuing. So how about non-vaccine strategies? It's quite clear that it's going to be a long time before we have a safe and effective HIV vaccine through any of these approaches that are being tried today. So meanwhile, there are several approaches for HIV prevention that are highly promising and will come to fruition long before there's an HIV vaccine. So for non-vaccine strategies, there are broadly neutralizing monoclonal antibodies and long-acting PrEP. So monoclonal antibodies for prevention is a concept that really came into being in 2021 when with this paper from the HVTN that showed that even a single antibody called VRC01 could give about 75% protection against HIV infection, but only if the virus is highly sensitive, which makes sense because a lot of viruses are resistant to particular monoclonal antibodies. And so clearly the clinical application of this concept is going to be to develop cocktails of antibodies. Also, the protection seen in humans with antibodies essentially correlated very well with the titers that were predicted to protect from non-human primates. So we think that the non-human primate model actually is a good model for the use of antibodies in humans for protection. But the future of antibodies for prevention will be antibody cocktails. And in particular, this is a cartoon of HIV envelope and the different antibody targets. And basically, if you combine an antibody against the main sites, the V3 glycan supersite, such as PGT121, the V2 apex site, such as PGM1400, and the CD4 binding site here, shown as, in blue, the VRC07 523LS, or their equivalents, would make a very good trivalent antibody cocktail. That would give about 99% coverage of global viruses and 83% coverage if you want to cover the virus with at least 2 antibodies in the cocktail. In addition, what's most proximate to clinical use is long-acting PrEP, long-acting pre-exposure prophylaxis. And this is a study we did with our colleagues from Gilead last year, in which we showed that a long-acting capsid inhibitor called lenacapavir, a single inoculation of lenacapavir gave 6 months of protection against weekly SHIV challenges in macaques. So really a very stringent challenge. And we were able to define the parameters and the doses that then led to failure. And this is in phase 3 studies right now by Gilead. Lenacapavir is recently approved for therapy, and then for prevention, we will know shortly whether these results in macaques are seen in humans. But clearly, long-acting PrEP is what's closest to what's closest to clinical utility today. And either the long-acting bNAbs or lenacapavir, if they prove to be successful, then they're modalities that could be given to patients twice a year, every 6 months. So it's very practical. For example, someone in college might go home for summer vacation and Christmas break, go to their doctor, get 1 shot of a long-acting capsid inhibitor, maybe a long-acting bNAb cocktail, and with 2 interactions with the healthcare system a year, they could be protected from HIV. Also, broadly neutralizing antibodies may have therapeutic potential, either as therapy, pure therapy in terms of ART replacement, or as part of HIV cure strategies in terms of ART-free virologic control. There's currently no strategy that I know of that can truly aim at true eradication of every single provirus. So we've explored this in the HIV cure space for a number of years now. And one of the studies that really made us excited about this was a study we published in 2018, in which we showed that the monoclonal antibody PGT121, which bound the V3 glycan supersite, together with an immune activator, which activated TLR7 called vesitolimod, was able to delay viral rebound following ART discontinuation in SHIV-infected non-human primates. So animals were infected with SHIV, they were put on ART, and then they were given antibodies with or without this immune activator. And then after the treatment period, then we stopped therapy and looked at viral rebound. Whereas 100% of sham controls showed rapid viral rebound, the immune activator alone didn't really do much. If we gave a series of infusions of antibodies, we were able to somewhat delay rebound. But if we gave the antibodies with the immune activator, then we were able to substantially delay rebound and in some cases prevent rebound. So this just shows the Kaplan-Meier curve from the same experiment, showing that we were able to substantially delay and or prevent viral rebound following discontinuation of antiretroviral therapy in macaques with PGT121 together with an immune activator. We then developed a clinical lot of PGT121 as a monoclonal antibody, and we brought this into a first-in-human clinical trials ourselves, and we showed that in viremic persons living with HIV, a single infusion of the antibody led to about a 2-log reduction in viral loads. However, then by about week 4, there was viral rebound in essentially everyone, largely with resistant virus. So this suggests that the use of broadly neutralizing antibodies as antivirals essentially it looks very similar to any sort of high quality single antiviral agent. Of course, you would not use a single antiviral agent in patients, but it looks fairly competitive in terms of potency to any known single antiviral agent. The problem with antibodies is that there's a high amount of resistance. So if participants had a virus that was resistant to the antibody, then of course we saw essentially no effect, which is actually a control saying that we need virus to be sensitive to the antibody for the antibody to work. Now, interestingly, in persons living with HIV who had very low starting viral loads, then the antibody had a profound effect. And 2 out of 4 individuals had this prolonged therapeutic effect that really could not be predicted by antibody kinetics, and as of today still remains unknown. So a subset of individuals seem to have a profound effect with antibodies that goes way beyond the actual durability and the pharmacokinetics of the antibody itself. We then developed a triple antibody cocktail I told you about earlier that involved. This is a first-in-human administration of PGDM-1400, as well as the triple cocktail of PGT-121, PGDM-1400, and the VRC07 CD4 binding site antibody. And in a therapeutic setting, we were somewhat disappointed in that we got about a 3 log drop, but still, even with 3 antibodies, we were not able to sustain viral control. This is just in 4 patients, but in all individuals, there was rebound afterwards. So we were thinking that antibodies are good at control, but they're not exactly what you would want to use in patients who are highly viremic. So we were inspired by this study that was done with different antibodies with Michel Nussensweig's group at Rockefeller, in which he used similar but different clones, V3 antibody 1074, which is like PGT-121, and a CD4 binding site antibody that's 3BNC117, which is similar to VRC07. And he showed that there is more prolonged viral suppression if you actually can if you can suppress people with antiretroviral drugs first, and then give the antibodies and stop therapy. So we actually designed a study and analogous to this, except using a triple antibody cocktail instead of a dual antibody cocktail. We reasoned that for drugs, it really was the triple cocktail that made the difference. So we wanted to see whether the use of a triple antibody cocktail would be better than a single or dual antibody cocktail in humans. So this is a very small study, 12 persons living with HIV on ART with fully suppressed virus. Then we gave 6 monthly infusions of this triple antibody cocktail at 20 mg per kg. And ART was discontinued in all individuals 1 day after the first infusion. And we monitored viral loads every 1 to 2 weeks with very strict ART restart criteria. So here is the time schedule. There were 6 monthly infusions of the antibody cocktail. ART was stopped as soon as we started the first one. The first question is, would we be able to maintain undetectable virus during the dosing period? In other words, could we actually use a triple antibody cocktail as ART replacement and get clinically relevant virologic suppression in at least the majority of individuals? The second question is, well, what would happen during the washout period? Here, we maintain people off of therapy, and we did not give any further treatment. So no ART and no monoclonal antibodies. And so we would expect that as the titers of the antibodies would go down, then we would see rebound. And at 44 weeks, it was the end of the therapy, and everyone would be recommended to go back on therapy. So the results showed that 2 out of 12 participants had an early rebound. We know that this is because they had baseline partially resistant virus in their reservoirs. But interestingly, 10 out of 12 participants maintained virologic suppression for the entire 24 weeks of dosing period. So 83% of participants in the study were able to replace their ART with bNAbs. Apart from the early rebounders, there were no other rebounders, and they all maintained suppression as long as their antibody levels remained therapeutic. By the end of the study, during the washout period, 5 more individuals rebounded in the setting of declining antibody titers. But interestingly, 5 individuals maintained undetectable virus for a full year, including 6 months after the last antibody infusion at a time when the antibody titers had declined to low or undetectable levels. So basically, the results of these 12 participants we found quite exciting. There were 2 that had early rebound. Then 10 out of 12, 83%, were able to maintain undetectable virus as long as antibody titers were therapeutic. So there was no induction of new resistance during this time period. And then the expected outcome is that individual would rebound as titers would decline, but 5 out of 12, so 42% of participants actually didn't rebound even after antibody titers declined and remained aviremic a full year after starting the study. And these participants were incredibly excited to come in for their viral loads, and every month they came in, they were just delighted not having to take pills. So why is this? We looked at the viral rebound, we looked at the viral reservoir size, and if we look at the different groups, the early rebounders, the late rebounders, and the individuals who didn't rebound at all, there was really no difference in reservoir size. So we don't think that the antibody directly impacted the reservoir. And we wouldn't expect that based on our macaque studies with that in the absence of any sort of immune stimulator. So of course, it was the antibody, the therapeutic activity of the antibody itself. Here's the PK of the antibodies. You can see this jagged sawtooth pattern, as you would expect for 6 monthly infusions of the antibodies, PGG-121 and PGM-1400. 3 individuals only received 3 infusions, the 2 early rebounders and 1 who elected not to pursue any further antibody infusions. The other individuals all had 6 infusions. There was no evidence of anti-drug antibodies. This is the same for VRC07, 523LS. You can see this antibody has a slightly longer half-life, so the decay curve is a little more slow. So we have the detailed virology on each of these individuals. I'm just going to show 2 in terms of time. This is one of the late rebounders, this blue individual. Basically had 3 infusions of antibodies. This is the individual that elected not to get any further antibodies. The only one, actually, who just chose not to. And then over about 6 to 9 months later, the 2 glycan antibodies, PGG-120 and PGM-1400, declined to below therapeutic levels. The VRC07 still had a low level. And at this point of time, this individual rebounded, and the rebound virus was completely sensitive to all these viruses. So this is an example of a rebound of a sensitive virus because there was no longer selection pressure present. How about one of the early rebounders? This one. The first rebounder rebounded just in about less than 2 months, very strongly here with very high levels of all 3 antibodies. And not a surprise, this individual rebounded with a resistant virus. Red is resistant, green is sensitive. And the resistance was against these 2 antibodies, 121 and 1400. It remains sensitive to the CD4 binding site antibody. And when we look back and we cloned out provirus from the reservoir of this individual, at baseline, we were able to detect that at baseline, this particular individual had virus that had resistance to 1400 and had at least partial clones, a subset of clones that were resistant to 121. So it's very clear in this case, this person was one of the rare people who rebounded early because there were at least some variants in the reservoir that were dually resistant to both of the most potent antibodies in the combination. And basically this story is quite concordant. The 2 early rebounders is because of baseline resistance. If you don't have baseline resistance, which we estimate to be in about 15% of people for this particular antibody cocktail, then you can be suppressed with a triple BNAb cocktail as long as you maintain therapeutic levels. So let me just conclude. The development of an HIV vaccine remains a global health priority, and novel vaccine strategies are needed. The Mosaic Ad26 envelope vaccine did not show efficacy, but Ad26-based Ebola and COVID-19 vaccines have been developed and deployed with important global health benefits. Long-acting BNAb cocktails and long-acting PrEP may be highly effective alternatives for HIV prevention as we await an HIV vaccine. Long-acting BNAbs may also have therapeutic potential either as ART replacement or as components of HIV cure strategies. And in a very small study that requires confirmation in a much larger study, we showed that a triple BNAb cocktail provided long-term virologic control in 10 of 12, 83% of persons living with HIV without the need for ART for the duration of antibody therapy. I'll stop there. Thank the large number of people. This is our group. This is a picture from our research retreat from January. It's the first time we were able to have an in-person research retreat for 3 years and a large number of organizations that were participating and collaborators in these studies. Thank you very much. I'll take questions if there's time.
That's great. Thank you very much. I will throw it over to the audience for questions. Dr. Jordan.
Thank you for a remarkable lecture. I was hoping you could comment a little bit on viral latency. First of all, is latency in the context of HIV true latency where there's absolutely no expression of viral antigens? And if so, are there strategies to try to reawaken those cells to drive them to a state where they'd be more susceptible to therapy?
Absolutely. So I didn't go over the basic biology, but when HIV infects, then the reason why it can't be cured with any drug regimen is because a fraction of the virus becomes latent. And latency is in resting CD4 T cells and essentially lasts for life. And the initial estimates that continuous antiretroviral therapy could cure you in about 50 or 60 years have now been changed to an infinite period of time, because there appears to be different decay curves of the reservoir, and after a period of time, it stops decaying altogether. And so basically, there is no treatment that exists today that can eliminate the viral reservoir and cure HIV infection, which is why ART treatment is for life. The question is whether the viral reservoir can be targeted, because for HIV cure strategies, if you want to cure HIV infection, you'll have to eliminate the viral reservoir. And so we and others are trying to develop ways to activate the viral reservoir with the idea that if the reservoir cells could be activated and awakened, then you could eliminate them. Because it's very easy with an antibody or a T cell or even maybe drugs to eliminate a cell that's actively producing virus. If the cell goes transcriptionally silent and you have a provirus sitting in a cell and the cell looks like a normal cell to the host immune system, with no viral antigen on the surface, then there's no way the immune system can target it. So it was initially believed in the early days that latency was absolute, that it was complete silencing of the HIV genome, which would essentially mean that it would almost be theoretically impossible to develop an HIV cure. Now it's believed that latency is really not absolute. There might be some cells in deep latency, but a large number of the reservoir cells have at least some level of viral transcription. And we think that's why that some reservoir cells might still be sensitive to potent CD8 T cells that might be able to kill a latency cell if it's just maybe transcribing and translating maybe a portion of the GAG gene and putting just one epitope on the surface. It might become susceptible then, even if it's not actively producing infectious virions. And the data for that is really some elegant sequencing data that really shows that the viral reservoir changes over time. And in many cases, after 10 or 20 or 30 years, the sequence diversity of the reservoir appears to go down rather than go up. And it seems to become dominated by clones. And those clones, sometimes they bubble up with just a little bit of replicating virus. And the clinical analogy of that is the fact that you sometimes see very low levels of virus that may not be replicating virus, but might be viral particles that emerge from these clones in the reservoir. And hence, if you look at an ultrasensitive assay, typical clinical assays are 50 copies per ml. In the research lab, you can get down to 1 copy per ml. You often can detect a little bit of virus. And as a clinician, we call them blips, and we generally ignore them because they have very little clinical relevance. So that is the clinical analogy to the fact that the reservoir on a molecular level essentially becomes more homogeneous over time with dominant clones. And that is believed to be the product of some low level of virus replication in the clones and cell division of those cells. So the reservoir cells are likely a large spectrum. There might be a subset that are truly in deep latency, but the bulk of them probably have some level or low level of transcription and translation, at least intermittently. And therefore, it gives us hope that cure strategies can target them.
I think we have time for one other question.
With the vaccine vectors, how many shots of Ad26 can you give to a person without losing its efficacy?
Sorry, how many shots before what?
how many shots of Ad26 vector can you give to a person without losing its efficacy because of generating immunity by the vector itself?
So, I mean, in this study, we gave 4. And if you give, for COVID, we gave 2. And you can certainly boost them about a year later. So if you want to give more than 3 in a short period of time, you probably want to have boosts on a different serotype, because you do generate anti-vector immunity with multiple shots. But you can certainly give 2 to 4 shots within a vaccine regimen.
Great. We are right at 1 o'clock. So Dr. Brook, thank you very much for this talk. If there are other questions, please come up.